So with that, I'm going to pass it off to Chris Chambers.
All right. Hello.
We can hear you.
Great. All right. Yeah. Hi, guys. This is Chris Chambers. Today I'll be doing a quick overview of the work being done on finfish in the mid-Atlantic states. If you have any questions, please hold those until the end of Chris Gobler's talk. By the way, Katie, right now my screen has my talk, but I see Chris Gobler on top of it in my panel for the webinar. Are you seeing my screen only, Katie?
I'm seeing your screen only, but because I chose the hide webcam option, I'm not sure why that's popping up. So
Gotcha.
if
Okay.
folks can see the webcam and they want to remove
Gotcha.
the little icon at the top that says webcam,
All right.
remove.
Okay. All right. I'll proceed. All right, folks. I first want to acknowledge our funders and contributions by our team. The Northeast Fisheries Science Center is where I'm located. You see here a list of collaborators and OA team members at the laboratory. The work is funded both by the NOAA Northeast Fisheries Science Center and the NOAA Ocean Acidification Program. And we also have contributions from interns, students from the Office of Education, the Hollings Program, specifically Jacob Strock and Raven Binko. So let me first start off by simply addressing the question of what is the problem with ocean acidification. In simple terms, there's too much atmospheric carbon dioxide as a result largely of the burning of fossil fuels. This increases the surface aqueous CO2 component, and that shifts the equilibrium of CO2 in the oceans to increasing carbonic acid, increase in bicarbonate, and a decrease in carbonate, and along with that a free hydrogen ion, which decreases the pH. This results in shown here a fairly consistent, with some interannual within-year variation, increase in atmospheric CO2, shown here in the purple color. This is from a field site at Mauna Loa in Hawaii. The seawater increase in CO2 also is increasing linearly over the last 30-plus years, and a predictable decrease in seawater pH at the same locale. This has resulted in a variety of consequences. It differs among species. I'll get into that in a minute, but it certainly may affect a variety of phenotypic expressions from otolith fish, life history features, as well as shellfish. And Chris Gobler will speak more about the shellfish responses shortly. We see here an example from pteropods of dissolution of shells and when placed in the high CO2 environment. And more recently, there have been a variety of studies showing that fishes of a variety of types, both from tropical to high temperate and boreal, are affected by high CO2 in terms of behaviors, fertilization rates, development, ontogeny, etc. So one aspect, if we can draw any generalizations about the effect of high CO2 on fish, is that it is life stage specific, and the young life stages are the most vulnerable. Our species of choice happen to be flatfish, and we focus on the embryonic stage, the young larval stage through the larval period, and into metamorphosis. I'll be speaking about that in more detail shortly. First of all, let me give you a sense of who's been doing what on ocean acidification effects, and more specifically, high CO2 effects in an experimental context on finfish in our region. There have been studies, a variety of excellent studies by folks in Stony Brook. Chris Gobler was one of the contributors to these studies, Hans Bowman and others, Chris Murray, his student, who are now at Yukon Avery Point, but the original work was done at Stony Brook on inland silverside, and also more recently Atlantic silverside. There's been additional work done by Denise Breitberg and her team at the Smithsonian Research Laboratory in Maryland. The folks at the Milford Northeast Fisheries Science Center Laboratory have conducted studies on scup, shown here, the green dot on the map in southern Connecticut, Milford, Connecticut. The team at Stony Brook also looked at sheepshead minnow, and we have been looking at winter flounder in our lab at New Jersey. We'll talk more about that in a minute. And both our lab as well as Tim Target's lab have been looking at effects of high CO2 on summer flounder as well. So in 20 minutes, I can't do justice to all these studies. What I'm going to do is focus on the kinds of results we've been seeing and make some general statements about the way people approach high CO2 or ocean acidification studies, and specifically looking at the biological effects in an experimental context. So what we've done, and the details differ from lab to lab, is we've evaluated a wide range of CO2 levels, and often we use a thermal regime as a co-stressor. Other people have used other co-stressors, and we'll mention that further later. Our approach has been, and this shown in this schematic, to provide our early life stages of fish with one of several, up to 5 originally, different CO2 levels or pH levels of the water, and up to as many as 5 different temperatures. The actual scheme is shown on the right here with our in our laboratory, where the vertical 10-foot PVC pipes represent 5 different CO2 levels. They're infused with different levels of CO2 from the canisters shown there in the foreground. When you look underneath that wooden loft, you see troughs, which are our water table, means the temperature control. And if you look in one of these troughs when an experiment's underway, this is what you see. Each one of these clear plastic tanks is a different CO2 level or replicate of that level within a temperature bath with an inflow and outflow of the targeted CO2 level. If you open up one of the lids of these containers, look down in it, what you see is something like this. In the center is with the float, air tubing those float around a plastic mesh coffee filter. That's where we incubate our eggs, and that's the size of container. And then we release the larvae into a larger 12-liter container, which is what the floating plastic bin is floating in. And on the right, you see outflow taps of 2-headed outflow. One's above the water line right now, one's below, and an inflow line on the left side of this container. And here's the kind of typical variation we see during the course of an experiment. Our nominal treatment levels are, of course, reported in this term, the actual CO2 and pH levels. They don't overlap. They're significantly different from each other, but there is within treatment variation. So let me talk about first our work on summer flounder, which is, again, being done at the Sandy Hook, or the Howard Laboratory of the Northeast Fisheries Science Center there at the northern tip of the Jersey Shore. Summer flounder, we chose it, one, because it's economically important in our region, very much so for the commercial fishery. It's different than the other species we work with in that it spawns offshore in the autumn. It has pelagic eggs. So the eggs in young life stages, larvae in particular, are experiencing the continental shelf waters. But the animals at flexion turn and start heading towards the shore and ingress into the bays either in late autumn or in the following spring after they're spawned in the autumn. Again, they spawn over the continental shelf. We happen to collect the adults when they're inshore in the summer during their feeding interval in our area. So our typical experiment for summer flounder, we have, in this case, we had used 3 CO2 levels. For our fertilization work, we also used multiple temperature levels. Typically, we have 3 replicates of each container, and depending on the study, 100 embryos or 300 larvae would be evaluated in terms of our design. The protocols are shown here: collecting the adult fish, in this case from a pound net fishermen in local waters, hold the adults in the laboratory and bring them in a spawning condition, and study their eggs and larvae and early juveniles. The response variables we'll be talking about today are shown here: gamete viability scored as fertilization rate, embryo survival, larval ontogeny, and I won't go into histology today. So here's the kind of results we see. On the left, I have the axis is fertilization success. That pertains to the various shades of pink to red to brown bars at each of 3 different temperatures, 16, 19, and 22 C, and in each of 4 different CO2 levels or pH levels as shown here. The right axis shows the percent difference from our ambient water of 7.9 pH. What we see here is that there is a strong negative effect of increased CO2 on fertilization likelihood. Furthermore, there is a CO2 by temperature interaction for summer flounder. And there's also as well as PCO2 by female or parental interaction from in this case the mother's contribution. And here are the summary ANOVA results for these outcomes. So again, high negative effect of high CO2 on fertilization rate in summer flounder with interaction with environmental variable temperature and also a parental contribution in terms of female source term. In terms of survival of the embryos of summer flounder, what we see is that at our, this is the all-at-1 temperature now, survival to hatching very dramatically, even from low to intermediate levels. About half of the 50% of the animals did not survive to hatch at intermediate compared to low, and another 50% below that at the highest CO2 level that we implemented in that particular experiment. In terms of development of larvae, we score a variety of measures based on anesthetized larval lateral images. Here are two pictures showing day 0 larvae and day 28 larvae. We know a lot of these variables that we measure, lengths and body depths, are going to be collinear. So we perform a PCA to reduce the number of dimensions from the principal component analysis to the manageable 1, 2, or 3 for our subsequent ANOVAs or MANOVAs. I want to point out that one of the differences as the animal increases in its development, not only does the body thicken, the mouth get larger, and so on, but you see back near the tail of a 28-day-old animal post-hatch, 2 yellow lines. That represents a quantity, the flexion angle, which is a precursor. Flexion is a precursor to eye migration, metamorphosis, and settlement in this species. So we scored flexion angles as an indicator of development. And here's a summary plot of principal component axis 1 versus 2. And you see that most of the measures are tend towards the right side of PC1, meaning a positive contribution to that axis. So they are collinear. We are going to be analyzing PC1 scores and also PC2. And you see the primary vertical direction variable is flexion angle, FA, in that plot. So these are clearly quite different measures of what's going on during ontogeny. Flexion angle representing development. Much of the other measures represent a more consolidated picture of body size as a function of age and treatment. And here are the kind of outcomes we see. Panel 1, A, B, and C show PC1 score. This means a body size measure, and what you see is that the animals are actually larger at hatching at intermediate and higher CO2 levels and continue to maintain that advantage at least midway through the larval period. As they approach metamorphosis, the higher CO2 level animals are decreasing in their growth rate, if you will, and in the bottom right panel, panel D, we're plotting PC2 score, i.e. development rate, that flexion angle I mentioned earlier. We see the high CO2 level animals actually more advanced developmentally, i.e. they're going to metamorphose earlier, and in this particular animal's biology, that means they will be likely ingressing earlier in the autumn before winter, based on the size trajectory we confer from these first PC1 scores, they're going to be metamorphosing and settling at a smaller size than animals at lower ambient CO2 levels. Our winter flounder work is a bit more layered. Winter flounder biology, of course, is different than summer flounder. All the experimental work is being done still at Sandy Hook, but they spawn inshore in winter in embayments and estuaries and release demersal eggs that stick to a substrate, and the larval duration is longer, 5 to 12 weeks in general. So they occupy a very different habitat, and we're contrasting those that spawn along coastal inshore estuaries in New Jersey with those that spawn along the inshore habitats and embayments in Massachusetts with each other. That contrast is one contrast, latitude, if you will. And we're also comparing those with some offshore spawning animals collected near Stellwagen Bank and applied or used in the same kind of experiments. And so what we did is implement a two-way design, CO2 by temperature, and again, the numbers differ depending on if it's the egg stage or the larval stage, or the replicas differ depending on if it's a gamete only or a larval period duration study. Same kind of protocol, collect wild fish, spawn them, monitor eggs and larvae. And here are the response variables that we're talking about here. Again, viability, larval ontogeny, and prey consumption. And we have a collaborator at UNH, Elizabeth Fairchild, and one of our grad students who are looking at the otolith structure and how it's affected by high CO2. Here's what we find in terms of fertilization success. I'm showing the results for the Massachusetts coastal population here. And in contrast to what we saw in summer flounder, the fertilization rate increases with increasing CO2 or decreasing pH at both temperatures. Summarizing that bullet, there's also a PCO2 by temperature interaction. and here, also a female interaction with PCO2 in the summary ANOVA results for that experiment. Similar but a bit different in the New Jersey coastal population, we still see an increase in fertilization rate at lower pHs, with a peak at intermediate values, if you will, around the 7.7 or so pH. We again see a female by CO2 interaction, so once again a parental effect on these responses. And here are the summary ANOVA source terms and p-values. In terms of the size of the animals, the larvae in these studies, these are stoag and bank animals, again the offshore spawning group. At hatching, the animals are larger at a higher PCO2 level, which is similar to what we saw for summer flounder. And they also happen to be smaller at high temperatures, which may be a compounded effect of climate variability on this organism in the brief ANOVA summary. In terms of settlement or metamorphosis in this case, what we find is that there's smaller size at higher PCO2 level, as was true for summer flounder. So they're metamorphosing earlier and a smaller size, all else equal. And there is also a temperature by CO2 interaction. Again, brief results for the ANOVA table. I want to mention another kind of result we've been scoring in the last year or so, and that is on prey consumption rate. There's one recently metamorphosed day 0 post-metamorphosis winter flounder in that bowl of, and within in that bowl as well are a large number of prey, in this case brine shrimp, that we use in our a functional response trial, offering individual flounder a known set number across a geometric series of prey abundance, scoring how many are consumed, and then fitting what's commonly known as the Hollings type 2 functional response, where A represents the slope at the origin, handling time, or the reciprocal of that is the asymptote, and we plug that into this equation to determine or estimate those parameter values. And what we find is that maximum consumption, which is the inverse of handling time, is lower for animals recently metamorphosed from a high CO2 larval environment. And the consumption rate is also lower at cooler temperatures. So again, the larval experience results in a lower consumption rate by these just metamorphosed juveniles. And again, in this case, another case example of interaction between another environmental variable. And the ANOVA summary is there. So let me just summarize our work, but touch upon some of the other results that we've seen throughout the region. In our case, elevated CO2 effects varies with responses measured, and this is not unusual, not just in our region of the country, but worldwide in terms of various species that have been evaluated. Fertilization rate decreased in summer flounder at higher CO2, but increased in both populations of winter flounder that we've evaluated to date. And there are predictably and regularly interactions with other variables like temperature and parental effects. The size of the larvae at hatching in both species was larger at higher CO2, but the ontogeny was accelerated, resulting in a smaller size at settlement and metamorphosis. And prey consumption in the one population we've evaluated this in shows that a high CO2 environment for larvae results in a lower prey consumption. That's corrected for body size, by the way, in winter flounder. So in terms of what's next, some of these points or suggestions are being addressed by some of the other researchers in the region, and currently, in some cases, in some of their publications. We need to proceed with a strategic increase of the species evaluated. Ideally, selecting representative species that are also ideally could be evaluated at multiple laboratories for interlaboratory comparisons if that is within the cards for us. We would like to broaden and unify the response variables considered. Of course, we want ecologically relevant response variables. Some folks are more direct their attention to mechanisms. Many of us look at the whether or not affected occurs, and we're interested in the shape and extent of that effect. Some of the regional investigators are looking at multiple stresses already, including dissolved oxygen by, again, Hannes and Chris, as well as Denise Breitberg and others, and our evaluation of thermal regimes and has already been underway. We would like, in my lab at least, to have a more complete assessment of the plasticity of the range of response to elevated CO2 and other stressors, so we can better quantify how these environmental factors affect the responses that we're interested in, and then plug them into models of those processes. Further, we'd like to have some sense of the adaptive potential in response to high CO2 and other co-stressors. And this has been addressed already in one paper, at least, by collaborators of Chris's in the Stony Brook lab, the MEPS paper. I'm thinking of Malfeasy, I think is the author's name. And as I mentioned a while ago, we'd like to quantify these effects and the likelihood of adaptive change so that we can put these into attribute-specific recruitment models, such as individual-based models. That was a quick run-through of the work being done. I'm sure I left out a lot, but I'd be glad to address any questions later if you have them after Chris speaks.
All right, thank you so much, Chris. I'm going to switch over now to Chris Gobler, who will speak to acidification impacts on shellfish in the mid-Atlantic. Quick reminder that if you do have questions for either of our presenters, to please include them in the questions box, and I will collate them for our Q&A after Chris Gobler's presentation. Chris Gobler, I think you might be muted.
Can you hear me now?
There you are.
Okay. How's the screen look?
The screen looks good, though. We're on. There we go. Now we're on the full presentation mode.
Okay, so let's go then. That was a great introduction by Chris Chambers, and I'll move right into bivalves now. And I'll just emphasize in this slide here the fact that we know that as CO2 levels go up, that results in a decrease in the availability of carbonate, and that's shown in this graph here as well. And therefore, with declining availability of carbonate, which is the limiting compound in the synthesis of calcium carbonate, we've known from the beginning that of ocean acidification research that organisms that are calcifying will be vulnerable to ocean acidification, and there's been many papers that have shown that. In New York, as an example, many of our calcifying organisms, many of our top fisheries are calcifying organisms, particularly filter-feeding bivalves. So this has a big, could have a big economic and socioeconomic effect in New York and likely other states in the Mid-Atlantic. Most of the research that we've done on bivalves has been on early life stages, just as Chris Chambers explained that the early life stages of fish are the most vulnerable. We've found the same thing with regards to bivalves. And so we've been spawning hard clams, eastern oysters, and bay scallops, as shown in the upper right video, and then tracking the performance of larvae, which is shown in the lower left. And one of our first studies is shown here from the prestigious National Academy of Sciences, looking at both bay scallop and hard clam larvae at multiple levels of CO2. And you can see pretty much a dose-dependent relationship between the survival of these larvae, and this is survival all the way through to metamorphosis with lower levels at the higher levels of CO2. And the one very particular interesting thing in this study was the fact that when we used near pre-industrial levels of CO2, we actually had better survival than at the time what was modern day levels of CO2, suggesting that we've already changed the ability of bivalve larvae to survive in today's ocean. In addition to effects on survival in this paper, we also showed that higher levels of CO2 resulted in bivalves that were smaller in their overall size. And when we cross-section these individuals, shown here are the hard clams, and you can see that it's not just their overall size, but also the thickness of their shell that's reduced when exposed to high CO2 during that larval stage. And so beyond survival, size, and thickness, other impacts of high CO2 on early life stage bivalves include changes or reductions in or alteration of shell morphology, reduced RNA to DNA ratios, slower calcification rates, slower rates of metamorphosis, and a lower lipid content. And all these things in an ecosystem setting will probably lead to additional mortality beyond what we already showed experimentally. I want to transition now to talk, just for a moment, a quick diversion to talk about what I'd call coastal acidification, and the idea that there's beyond the CO2 that we know is coming from the atmosphere. We know in coastal ecosystems there's many other sources of CO2. And we know, for example, that most estuaries are net heterotrophic with a consumption of oxygen. And therefore many coastal zones have been shown to be hypoxic. And one study that we recently published has emphasized the idea that when you do have coastal zones that do have eutrophic, high levels of nutrients, that the same conditions that lead to low levels of oxygen also create acidification, high levels of CO2 and low levels of pH. And so we've been looking at this as in multiple sites in the Northeast and also in the Mid-Atlantic. And I'll just show here one of the first examples of this, looking through Jamaica Bay, which is in the southeast corner of Long Island, part of New York City, and essentially showing that as you go, these are surface waters, as you go from the back end of the estuary out to the ocean inlet, you have an increase in both oxygen and CO2, but that you have very high levels of CO2 back in the estuary along with low levels of oxygen, and there's a close correlation between these parameters. This is a more recent look at that same system, Jamaica Bay, and this is a tri-data plot. So it's looking at pH and oxygen with data points colorized to show different levels of chlorophyll, which is phytoplankton biomass. And essentially what it shows is the strong role that algal blooms and phytoplankton play in regulating the pH and DO in this system. When we get the high levels of algal biomass or a phytoplankton bloom, that drives up pH and oxygen. And I should mention we're looking here at thousands of data points, of course. This is continuous monitoring in the system from May to October. But when you don't have an algal bloom and you have lower levels of chlorophyll, you can see the system just sags to these very low levels of pH and oxygen. Beyond Jamaica Bay, we've also looked at this in Long Island Sound and seen that during late summer, this is a section plot going from on the left, New York City, on the right, out to the middle of Long Island Sound. I should mention Long Island Sound by volume is the third largest estuary in the United States. And that during late summer, when low levels of DO, dissolved oxygen, develop in the western part of the sound, closer to New York City, we can currently see that most of the estuary has low levels of pH, very high levels of CO2, particularly in bottom waters, and is undersaturated with regards to aragonite. And I'll emphasize that the CO2 levels that we see in these bottom waters are higher than the levels we expect for the open ocean by the end of the century. And this is a seasonal progression that occurs. So I showed late summer, and what these plots here show is oxygen and pH, and showing how as you transition from spring into summer, that's when the low oxygen and low pH conditions develop, and then they go away when you have the overturn of the system headed into the fall. But I will note that acidification starts earlier and lasts longer and can be more widespread than the low oxygen conditions. So knowing that these coastal estuaries are both acidified and hypoxic, the next thing we want to address now, going back to the bivalves, is what are the implications for early life stage bivalves. And to look at this experimentally, we first looked at the literature and noted that most experiments looked at this simply by bubbling nitrogen gas, which does a great job of creating low oxygen conditions. But what we suspected is that high level of nitrogen, or bubbling in nitrogen, is also going to drive out CO2. And therefore lead to low oxygen, but also low CO2 and high pH. And I just showed you in the previous slides that that's a condition that does not co-occur in an ecosystem. So to look at it another way, when you bubble with nitrogen, you drive up the pH, but just looking at observations that we made a few summers ago in a coastal estuary here in New York, the conditions are that as you have low oxygen, the pH goes down. And again, that's because of respiration, producing CO2, consuming oxygen, and driving down the pH. So while we've had decades and decades of research into hypoxia, all of that research, almost all of it, has been done not only not considering pH, but actually creating a pH condition you don't find in the ocean. So we've begun a system whereby we're bubbling experimental chambers with nitrogen gas, but also with CO2 and air to create a condition that we do find in ecosystems. And that is a condition, if you look in the red lines here, and this is just showing data from an experiment, of both low DO and low pH, which we actually find in an ecosystem. And of course, we're able to do a full factorial experimental design by also just altering just the CO2 or just the pH to isolate the effects of these two different parameters. And so when we've done this with larval bivalves, what we found is that different pH and DO have different effects on different attributes of the larvae. And so as I showed you before, this is the survival of larvae exposed to either low oxygen, low pH, or both conditions. And as I showed you before, it's the pH that's driving down the survival of bivalve larvae. And that actually they're fairly resistant to low levels of oxygen with regards to survival. However, if we look at the size of the individuals, we see a completely different story. There's a slight knockdown in their size with regards to the low pH, but it's low oxygen that really drives down their size and keeps them very, very small. So, both of these parameters are important, and if you only looked at hypoxia, you wouldn't get the full story, and the same is true for pH. And we know in an ecosystem setting they're having both low oxygen and low pH. Now, we've also, I've emphasized early life stage organisms and specifically larvae, and that's because the older these bivalves get, the more resistant they are to these conditions. But in an experiment here with juvenile hard clams that are 4 months old, in this same paper that the other data I just showed was in, we found that those individuals were resistant to low oxygen or resistant to low pH. But that's if they experience those parameters individually. But if they experience them concurrently, as happens in an ecosystem setting, you get a synergistic effect of reduced growth rate of these hard clams when they're experiencing both hypoxia and acidification. One other attribute of low pH and low DO that I want to emphasize is that in these shallow estuaries, these acidification and hypoxia conditions actually vary on a diurnal basis. And that is because at night, without any photosynthesis, you have the pH and DO being driven down, whereas during the day, photosynthesis is actually doing the reverse. And so this is a paper with Hannes Baumann as the lead author on the North Shore of Long Island, showing that tight correlation between pH and DO, and the fact that at night you see the levels drop and during the day they rise up. We've also seen in other places now here going back to Jamaica Bay. These again are section plots, but in this case the x-axis is hour after sunset. And so, if you look at the beginning of these plots, you can see it's already stratified, and that, again, the bottom waters do have low pH, low DO, high CO2, undersaturated. The surface waters are a refuge, however, because of photosynthesis. But as you transition to night, you can see the whole water column starts going towards hypoxic and acidified conditions. So just emphasizing, again, the day-night variation in these parameters. And so recently we've looked into how these day-night changes, or these diurnal changes in CO2 and dissolved oxygen may be affecting early life stage bivalves. In fact, published 2 papers on this in the last few months. One is a featured article in Marine Ecology Progress Series. And so what we've been doing is essentially using timers and gas proportionators to mix the gases I already talked about, deliver to experimental chambers, and look at the same sort of experimental designs I described before, low pH on its own, low DO on its own, and both, but looking at it both being delivered chronically, so steady level of low pH, low DO, and also diurnally, and that's where the timer comes in. So during the day, we're delivering just normal levels of CO2 and oxygen, and the acidification only happening at night, mimicking what's going on in an ecosystem setting, and that's what's shown here. Again, the variation in pH and DO between day and night. And so this is a busy graph, but to give a sense of what we see when we do this, this is the survival of bay scallop larvae, and underneath you can see the numbers are the conditions that we're setting up. So in this particular experiment, the diurnal fluctuation in pH or DO or both conditions resulted in either pH or DO conditions that were pretty much halfway between the control and the low conditions. So for example, the low pH was higher, the diurnal pH was higher than the low pH because it's fluctuating between the ambient and low conditions between day and night. And the same being true for the low oxygen and both conditions. But despite the fact that these diurnal treatments have conditions that are significantly better than the what we call low conditions, because they're cycling again between high and low, when we then focus on the performance and actual survival of the larval, in this case scallops, what we see is that their performance is no better than if they had experienced chronically low conditions. And so, the thought had been that by experiencing good conditions by day, the individuals may actually be just fine. But in these conditions, what we actually are seeing is that the large fluctuations between high and low levels of pH and DO are actually leading to outcomes that are just as bad as if they had chronically low levels of pH and DO. In another set of experiments shown here, we actually contrasted two chronically low levels of pH, what we call low and reduced level, with a diurnal fluctuation between the control condition and the low condition, which would give a mean the same as what we call the reduced condition. This experiment is showing for, on the top, larval scallops, on the bottom, larval hard clams, and in both cases, the diurnal condition that fluctuated between ideal and low conditions that had survival rates that were really no different than that of the low condition, and actually were worse than the what we call reduced condition, even though the chemistry on average for the pH and DO was the same. Again, affirming what we showed in that last experiment, that it's the fluctuations that seem to be having an added negative effect beyond just the absolute levels being achieved. Okay, and for the last set of data that I want to show, I'm going to again go back to Jamaica Bay and to show some experiments where we actually deployed bivalves into the bay where we know there are these hypoxic and acidified conditions, to understand in an ecosystem setting how might this be affecting the outcomes. We specifically in these experiments used juvenile stage oysters and juvenile stage mussels that were deployed in cages. And so the first experiment I'm going to show is from June. This is actually 2015. And this is a point in time where actually the conditions weren't too bad in Jamaica Bay. Again, I've emphasized earlier that if you get algal blooms, which you can sometimes get early in the summer, they can actually drive up the DO and pH. And that's what we saw during our June experiment. Conditions for CO2 and DO that were not all that different amongst the stations, and actually not acidified or hypoxic. And when we looked at the growth rates of both eastern oysters, in this case blue mussels, what we found is that the performance was a little bit better back in the bay, and this tracked essentially with food. That is to say, there were higher levels of suspended food particles in the back part of the bay, but we had fairly good growth rates across all stations, against slightly higher in the back part of the estuary. We then performed the same experiment later in the summer, and this is the time when we typically see the collapse of those algal blooms, and therefore very high rates of respiration, which lead to both hypoxic and high CO2 acidified conditions. And that's what was seen during the September experiment. And you can see that the worst conditions, the further back in the bay you go, the worse conditions get. And I should emphasize that JFK is literally John F. Kennedy Airport that was built on a salt marsh within Jamaica Bay. So our one station is off of a pier that's right off of the airport. And then the other station, the furthest station from there, is the Rockaway Inlet. And you can see in this particular experiment that was the station that had the best conditions with regards to both dissolved oxygen and levels of CO2. And when we look at the performance of the mussels and oysters in this particular experiment, we see actually a full-on reversal of the conditions compared to what we saw in June. And that is back in the bay by John F. Kennedy Airport, where the levels of oxygen were low, levels of CO2 were high. We had a significant reduction in the growth rates of both the oysters and the mussels. And I should say, or I should emphasize, that statistically there were no variations in temperature or salinity amongst these sites, so the conditions were within the ideal range for bivalves, and that the food availability was actually higher for the JFK site in the back part of the bay. So leaving us to conclude that the poor conditions with regards to oxygen and pH and acidification were likely impacting these bivalves. Okay, and so to emphasize some take-home points here, I've shown you that there are northeast estuaries that experience hypoxia and acidification together, specifically during the seasons when we know that these early life stage bivalves are spawning and developing. So the overlap is unfortunate from a life history point of view. The levels of acidification that are occurring within the estuaries are shown to both additively and synergistically depressing the growth and survival of early life stage bivalves. And when they fluctuate diurnally, can actually create conditions that are actually worse than chronic exposure to the same exact chemistry. And so it may be that both hypoxia and acidification are combining to affect production of fisheries in coastal ecosystems. And then just a final concluding point is that both federal and state standards for protecting fisheries in coastal ecosystems with regards to general water quality typically focus solely on oxygen and certainly do not consider pH. And so the fact that these two parameters fluctuate together and are creating conditions that together are affecting the outcomes of these individuals could mean that we're leaving our bivalves and maybe even finfish more vulnerable than we recognize to eutrophication-driven modification of water quality. So with that, I will end, and I think we're on to the questions, but if we don't get to questions and anyone would like to email me, my email is there, and I would welcome questions on email as well.
Great, thank you so much, Chris. We do have a couple questions to start us off here, but for attendees, please do continue to send those questions in, and I'll address them as many as I can as we go along. So the first one I have here is directed towards Chris Chambers, but of course, if either of you want to chime in, please feel free. And the question is, do you have thoughts on why you see different responses in summer and winter flounder?
Right. Hi, this is Chris Chambers. Well, I do. I mean, our hypothesis going into the work was we expected that the summer flounder, given its early life stages, are in more stable habitats, if you will, in terms of average as well as variance in CO2 and other parameters. They'd be more sensitive to the challenges we're subjecting them to than winter flounder, which experience, again, estuarine environments during their egg and larval stages. That's one of the reasons why, by the way, we wanted to contrast the stellwag and banker, the offshore winter flounder, with the inshore animals to get a within-species contrast of populations from two distinct habitats.
Great, thank you, Chris. Our next question is for Chris Gobler. As you have said, Long Island Sound and other estuaries have hypoxic, hypercapnic conditions, and yet bivalves still persist in these areas. Do you think that bivalves in the wild are acclimated to these conditions more so than those spawned in the laboratory? Or could it be that wild stocks are quote unquote just hanging on, and that the present populations we see reflect the lower end of what is possible if estuaries experienced higher DO, lower CO2?
I think it's probably a bit of both. I mean, just for perspective, in New York, and since the mid 20th century or so, landings of mussels, scallops, oysters, and clams are down between 70 and 90%. Now, some of that we know is due to harmful algal blooms, but I have a paper in review at Marine Ecology Progress Series looking at taking our experimental results and then trying to translate those into modeling full population results. And they suggest that the CO2 changes may be responsible for some of the reductions in fisheries production that we're seeing. But we can't discount also that there could be some adaptation, and that there's probably some phenotypic plasticity in these populations, and perhaps some of the weaker lines that are less robust against things like acidification, hypoxia, through the years have been weeded out. So I would think both. I believe both things may be going on.
Great, thank you. This one's also directed towards you, Chris Gobler. It says, why do you believe that diurnal fluctuations of dissolved oxygen and pH are worse for bivalves than chronically low DO and pH? Could it be that a physiological stress is responding to rapidly changing environmental conditions?
Yes, and of course, first thing I'll say is that these are complex experiments. And we're really only looking at we're looking at realistic changes, diurnal changes in pH and DO. That is, the levels we're actually seeing in a place like Jamaica Bay. But of course, probably part of the result will be the extent of those fluctuations. So under the extreme fluctuations that we actually do see, yes, the outcomes are worse. Now, bivalves do have something that they go through, particularly juveniles, known as anaerobic respiration, which is a whole different set of biochemical reactions. And it may well be that it's switching on and off between going through aerobic and anaerobic metabolism is just a very large cost, and that these bivalves, are better off if they, getting to, say, anaerobic respiration and then just carrying that out, as opposed to having to swap between the two. So I think I'll leave it at there. And, say also that I mentioned these two papers came out just in the last few months, so I think there's a lot more to be done to better understand this.
Okay, thank you. That's actually all the questions we have. So unless I have some last minute questions coming in here, I'll just thank both of our speakers, Chris Chambers and Chris Gobler, very much for presenting for us today. And thank you all for attending, and a reminder that our next webinar will be on February 21st. Thank you so much.
Thanks, Katie.
Thank you.